Okay, we're going to get things started, everybody. Thank you again for attending. My name is Joe Ferriolo. I'm the At-Line Director of Bioanalytics Applications for Repligen. I'd like to introduce our speaker, Jay West. Jay is a principal scientist at Bristol Myers Squibb in the Process Development Analytics Group in Devens, Massachusetts. He received his BS in Chemistry from Rensselaer, MS in Chemistry from University of Albany, and worked at Regeneron Pharmaceuticals for 2.5 years. Jay received his PhD in biochemistry from Boston College and performed postdoctoral research at Northeastern University. He started at BMS in 2014 as an analytical scientist. His focus for the past 5 years has been on process analytical technology, particularly on applications for downstream biologics process development. Today's talk is biologics concentration measurement using the inline FlowVPX instrument during the UFDF process in drug substance manufacturing. Our speaker, Jay West.
Hi, can everybody hear me? You can just give me
all set.
Message. Great, Joe. Okay, so we're good. All right, I'll go ahead and start. And if there's any issues, I think that Joe is going to put it to me in the chat. So I'm going to get started here. And thanks very much for the introduction, Joe. So like everything he said was true, of course, and I've been at BMS since 2014. Enjoyed my time very much there. Let me go to the next slide. Okay, so just getting started here. All right. So we're on slide number 2. Okay, so let me talk about the meeting agenda. And the biologics manufacturing process, including what we call process analytical technology. That is, we call it PAT for short. And I'll describe what that is without going into too much detail. But it's basically instrumentation that is embedded into the process for the point of taking analytical measurements real time, as opposed to removing a sample and testing it offline. So UFDF stands for ultrafiltration, diafiltration, and it's part of the biologics process. And I'll show you in a diagram on the next slide where it appears in the process. And the potential benefits of an inline A2A monitoring instrument. An inline A280 value proposition. So when we're talking inline A280, I'm also talking about this FlowVPX instrument, which is, of course, why we're here with Repligen today. So the inline A280 instrument is the FlowVPX that we're going to talk about today. So at Bristol Myers Squibb, we've been doing UFDF studies with FlowVPX going all the way back to 2017 in the pilot lab. And I'm only going to talk about the last few years of those experiments, but suffice it to say, we have many years experience doing these studies with first the FlowVPE, then the FlowVPX instrument. Second part, so since we're now using the FlowVPX for the past few years, we've been seriously considering moving it from the pilot lab into the GMP space, because this instrument, the VPX, was built as a GMP caliber instrument. So we've been doing bridging experiments in the pilot lab, and we'll show you a limited amount and known gaps and challenges. And then the future potential for UFDF process automation. One caveat, or I guess one apology, or it's not my fault necessarily, is that we can't show you some of the single-use flow cell experiments that we've done this year because of some proprietary information between us and Repligen. But suffice it to say, in the future, we'll have that that information and data will be made public, which is interesting. But I'll show you up until basically all the data we have up until the last few months, and it's an interesting story. Go to the next slide. Okay, so what I'm showing you here, so I'm really focusing at BMS on the monoclonal antibody production and the biologics manufacturing process. I know now in biologics manufacturing, things have, and just even in the last 5 years, spread out tremendously into vaccines, into gene therapies. But at Bristol Myers Squibb, since I have been there, just like I said, starting in 2014, our focus has been on fusion proteins and monoclonal antibodies as far as biologics manufacturing and approved drugs that we manufacturing now. We have several monoclonal antibodies that we manufacture commercially. And this is, so it's evolved, like I said, starting about 20 years ago, I think, when I would maybe date the modern monoclonal antibody process, when they came up with protein A capture. That was a significant advance to have an affinity column for monoclonal antibodies. And of course, they are a huge share of the market for biologics. So let me just go into what has evolved at BMS, especially, into a platform process, so that we now can sort of plug and play with a monoclonal antibody directly into this platform and come up with a nice pure product at the end. So as I mentioned, protein A was a resin that was created back in, I believe, the 1990s, and now it's commercially available, which is extremely efficient capture of monoclonal antibodies. And I show two columns here because we use at BMS what's called a multi-column chromatography system. So that's actually something that that's only been in use about the last 5 years, and that allows you to use much less protein A resin. But I'll get on to what we're here for in the talk too, which is the UV-Vis. So after the protein A capture, there's a viral inactivation step, and then it has to go through polishing columns. So in almost all manufacturers of monoclonal antibodies use 2 polishing steps. Some use only 1, some use 3. And the polishing would be AEX, CEX, or HIC. After that, you do this viral filtration. Then it comes to the UV-Vis, which is the topic we're going to talk about today. And as I've shown, this is just a cartoon of the UV-Vis process, which I'm going to describe in the next slide, with a VPX in line. And this would be what the VPX's output would be, which would be concentration measurement over time. So you have a UF1 point, a diafiltration. So the diafiltration literally is exchanging the buffer that was in it, the protein was in at the beginning of the UV-Vis process, which is the viral inactivation, filtration, and then into the formulation buffer. So once the diafiltration is finished, and I'll talk a little bit more on the next slide about it, the concentration is increased to what's called this ultrafiltration 2 set point. And then it is recovered. And it's recovered into bags and then diluted to the final drug substance concentration and formulated. Typically formulated with PS80 and something like a chelator, like EDTA. Let me go to the next slide. Okay, went too far. Okay, so back to, sorry, I'm having some issues with my, okay. So here we are, slide 4. So I want to just describe the ultrafiltration diafiltration process to people who are not familiar with this manufacturing step. So this can be done on any substance, but typically it is done like on a macromolecule, like a protein, where you want to exchange the matrix and or concentrate the material. So you have a bulk, which would be your protein material. So typically at the beginning of a monoclonal antibody, you have to process the material. As you can see on the concentration plot on the right, it's about 10 grams per liter. So the first order of business is it's concentrated up to about 60, to 80 grams per liter. Now I'll talk briefly about concentration here too. At BMS, our drug substance concentrations are going up a lot over the past several years. And it used to be that typically drug substance was, the target concentration was 10 to 50 grams per liter. The reason that we're going to 150 to 200 now is because of subcutaneous injection. What that means is that monoclonal antibodies do not have to be given intravenously necessarily anymore. They can be done with a subcutaneous shot in the shoulder, which is far easier from a clinical perspective. especially these oncology type drugs, the immuno-oncology drugs specifically, which is one of the staples of our portfolio. So to sort of get back to concentration, so that's a clue as to why the VPX and PAT is especially important, an important tool inline A280 for UFDF, because you're concentrating to a very high concentration. So 200 grams per liter is approximately 20% by weight protein. So at that point, things are getting very crowded, things are getting very viscous. And I'll talk a little bit more about product quality issues and why it's really valuable to measure your protein concentration at the end of diafiltration and up at this ultrafiltration phase. Okay, now we'll get a little bit of a picture of what it looks like, not just as the cartoon, but how it looks like in the laboratory. And we have 3 different scales that I'm going to talk about today. One is bench scale, and I think did not include any pictures of that. But suffice it to say, this VPX instrument that we're talking about is scalable. It can be used in bench scale, and it can be used in the pilot scale. And that is what's shown at this lower left picture here. And you can see it sitting on a cart with a computer, and it's connected to those hoses. That's actually the bulk retentate bag. And what we have here on the right is that's another kind of actually a cartoon sort of. It's a cartoon over a picture, and that picture that I'm showing here is a UFDF skin, and that is in what we call the single-use facility here at Bristol Myers Squibb in Devon, Massachusetts. So I didn't mention too, our site is in Devon, which is about 30 miles west of Boston. It's a great site, I think, particularly if you like manufacturing biological drugs. We have a commercial facility for manufacturing monoclonal antibodies, and we have a clinical facility. And so that's today where I'm sort of leading up to where we're going with this VPX here at BMS in terms of the PAT landscape, that we intend to move this into a GMP facility. And the first GMP facility would be the clinical space, because commercial space, the change control is much more significant. So being a conservative business as biopharmaceuticals are, we're going stepwise into the GMP by first going into clinical manufacturing. So let me talk about why the VPX is a really potentially valuable inline A280 instrument. So the current, like I said, I'm speaking specifically for BMS. Almost all of our platform monoclonal antibody manufacturing processes go now up to 200 grams per liter. They are then subsequently diluted into the 150 to 180 gram per liter range. The inline FlowVPX dynamic range has been claimed all the way to be accurate all the way to 300 grams per liter. And we have some evidence that it goes actually close to that, but since we don't intend to go in our manufacturing processes that high, it's not really significant that we tested that high of a concentration. One of the great aspects of this inline flow meter, if I may call it that, is that it has potential as a platform tool for inline A280 measurement across the entire UFDF operation. So that's one of the reasons why we've come to respect and use the FlowVPX, is it because there's no limitations, there's no model building either. You just basically, it's pretty much plug and play. I'll talk a little about how you do method development and a little further into the talk. Okay, so this one is an animated slide. So A280 testing, I want to talk about how it's typically done during the UFDF process in the pilot and GMP scale. So that's one of the things that we didn't talk, I haven't mentioned yet, is that it's critical actually that you measure the A280 during the UFDF process at certain parts of the process, that UF1 point, the UF2 point. And that is done right now. It can be done at line or offline or inline. Okay, so here's the complete picture. So let me just we'll talk about clinical manufacturing, just to be clear here, because like I said, there's a couple different facilities and that we're talking about, clinical versus commercial. But in a typical clinical manufacturing, at the end of UFDF, they remove a sample. This would be the UF2 point. This is the critical one, because they have to know if they're within a certain target range before they can move on to do the formulation. So what happens is that right now, if QC lab does this operation, so testing is done post completion of UFDF, number one. So basically, this is a critical point, and the QC does the test. The reason QC is, they are considered, the gold standard of analytics. So they do the test because if you don't meet that target drug substance concentration, you could lose your batch. So they do this test very carefully. And of course, a handicap of having a QC lab do it is it requires manual handling of samples with a chain of custody. The result turnaround takes at least 2 hours. That's actually a very conservative number. It typically takes far longer than that, typically 4 to 6 hours, because of the sheer amount of paperwork involved and the number of people who need to review that. So right now, one of the problems that they're having, I guess it's a certain problem. As of now, we are manufacturing high concentration protein in the clinic without using a FlowVPX meter and without testing on the floor. I'm not going to talk about solo VPE at-line testing, but suffice it to say that is part of the overall strategy we're looking at BMS for basically doing, incorporating process analytical technology to accelerate the UFDF process to streamline. I'll talk about more the benefits in the next few slides. So value proposition. So this is something that's important. I know this isn't necessarily, well, I will talk about some science engineering, but this is actually most important to those of you in the audience who basically go to your management and say, we want to go and start using this in our pilot lab and our clinical manufacturing. They're going to say, why? Why can't you just pull a sample and go test it, say with your solo VPE or have QC? So the reason is because it's wise to incorporate inline A280, particularly is to decrease the process variability and product loss during UFDF and recovery. Without measuring the concentration in line, I may need to stress this point a little bit later on, but there's a holdup volume, and when they measure concentration during UFDF, the way they know when to stop at a UF2 point is based on mass, and because they take a concentration at the beginning, so based on the change in mass, they can extrapolate what the concentration should be, but they do not know what the exact concentration is. So therefore, because you have a bag and there's a holdup volume, the holdup volume is not being weighed. So that contributes to potential error in the estimation of your concentration at the UF2 point. So that's why measuring it an inline measurement of the entire bulk solution of the system is much more intelligent and accurate way of doing it than based on mass. So the reason you don't want to excessively concentrate, and it's good to know what the exact concentration is that when you exceed 200 grams per liter, which is possible if you, like I said, with a mass-based system and you're not directly measuring it, there's a potential for turbidity and extreme viscosity, because viscosity increases in a non-linear fashion, and it becomes literally almost like honey. So the difference, say, between 230 and 240 grams per liter is quite significant. So therefore, it's best if you can keep your controls very tight during a process such as this. Let me just take a quick break. So again, you want to increase process data and consistency. So that's something else that it's almost an intangible, is that you are creating a record of the process concentration for the entire process. So you have that nice portrait of what happened during the entire process from a concentration perspective, which you would not have without an inline Eliminate, at some point, taking offline measurements. Enable, so this is something that we're working towards, enable real-time forward processing and in-process release, potential alignment of technology to entire process and across manufacturing sites. So that's sort of a holy grail in a sense, is that if everybody was using a similar technology at your manufacturing plants, that can make tech transfer of, say, release methods for A280 concentration much simpler, because maybe you're all using a VPX and solo UV-Vis system. And that makes things very simple, as opposed to a gravimetric A280 based system. So how do we do the method development for variable path link extension? So that's what the original VPE and now VPX what meant, is that it's taking, I'm not going to go too much into this because, like I said, this is a whole other topic, but suffice it to say, it's necessary to understand how this technology works in order to use it. And it's based on the same technology that the solo VPE uses, where it samples there's a fibrate that moves up and down into a cell with a detector underneath, and it's shining typically using 280 nanometer light, which we would use for proteins. You would use something else for other substances, but we use 280 nanometers for proteins. So then you need to use at least 6 points to get a good slope on a line. So what you do is you're measuring the change in absorbance versus the change in path length of the cell. And in that, you can plot the line and extrapolate what the concentration is, of course, knowing what the extinction coefficient is. So from that, that's based on Beer's law. And in Beer's law, all the way up to about 2 absorbance units, things stay linear in terms of path length versus absorbance. Above 2 absorbance units, things start to go basically tilt. It becomes nonlinear. So if you're trying to fit a line to something like a curve, so if you look at the middle plot, this is actually from a solo VPE study where we were trying to determine the right number of points to use on the curve to get an accurate concentration. I'll kind of, since it's how we're doing for time, we're 11:26. I'm going to just basically say quickly that the solo VPE study, and this is confirmed by another group at BMS, that what happens is that if you at path lengths above 30 micrometers, for this particular protein, which actually has this monoclonal antibody, has a high extinction coefficient, the curve becomes nonlinear. So the best thing to do was to cut off the number of points to 6. And 6 point is accurate over a wide range of concentrations, and therefore it's the best compromise. So in lower concentrations, it's arguable that maybe a 10-point fit would be more accurate, but we've found really that 6 points works for all molecules. So we started using the VPX over a year ago, and we did first initially bench scale studies. We wanted to go up to very high concentrations because one of the limitations of the previous instrument, the VPE, was the dynamic range. Getting to 200 for certain substances, we had found that the accuracy was limiting. So what we did in these initial studies when we were screening the VPX was that we took this monoclonal antibody, the one I had mentioned with a very high extinction coefficient, and ran it and compared it to 2 other offline methods. So as I mentioned, the solo VPE before and the A280. The A280 is, I mentioned, a gravimetric-based method. When I say gravimetric, it means that you need to it's done in a cuvette, a quartz cuvette. And in order to get into the range where the 1 centimeter quartz cuvette, you need to dilute about 400 times. And that cannot be accurately done with a volumetric pipette. You need to use a gravimetric type assay, which basically means you're weighing it out on a microbalance. In any case, that is considered the gold standard of accuracy, that gravimetric assay. And that is what's used by our QC group to release drugs and to do some of the in-process measurement. However, solo VPE, we have found it's been qualified up to 240 grams per liter. And that was for this substance. And it's been found, actually, we've done a lot of tests that up to 240 grams per liter, the solo VPE is accurate. Above that, actually, it starts to run into issues. Whereas the VPX actually is accurate we found up to 260 grams per liter. There's a little science behind that. Again, I don't want to get into all the technical details, VPX actually has a smaller step size that it can use for the path lengths it uses. So therefore, whereas the solo VPE can only, the minimum path length is 5 micrometers, and it goes up in 5 micrometer path step sizes. The FlowVPX, I think, can go below. I'm not sure what the actual reading is because it's pretty short at that point. Below 5 micrometers, and it can go up in 1 micrometer steps. So that effectively increases the dynamic range considerably. So how much testing have we done to determine if the VPX is accurate at high concentration? Because ultimately, that is one of something that is absolutely critical if we're going to be using this as a high concentration detector. And the answer is we've done lots of studies and 6 different monoclonal antibodies we've used to assess the accuracy of the inline VPX. So here is just a table. I just put bench and pilot scale. So the bench scale would be like a few liters of say, like material that you're concentrating down, and that you would have a final volume of a few hundred milliliters. The pilot scale, which I showed you a picture of before, typically the volume will be about 4 liters. When you get to a clinical scale, that's actually 4 times more. So that goes up to about 20 liters of material at the end. So just wanted to let you know what kind of scales we're looking at. But in any case, we found, at least we have not tried the clinical manufacturing scale yet, but as the bench and the pilot scales, the VPX gives accurate data. And I only show actually comparison from the solo VPE offline reading with the inline VPX reading. And in all cases here, we found it to be within 4%. And I say within 5% because that's really the gold standard, is being within 5%. So that's obviously very good news for those who are considering using the VPX inline and want high accuracy. This is something that I intended to report on more, but we, like I said, because of some legal limitations, we could not show you the data. But I'll show you at least that in what we have been testing now that we know that the FlowVPX is accurate, the next step is to put it into the GMP facility. And I'm going to talk about some of the things, the hurdles for that. But I wanted to show you some pictures first, because one of the more exciting parts of, I find exciting because I like working in the lab, we're getting to test out these single-use flow cells. And I know Repligen, this is still a work in progress. It's being developed. Like I said, we're kind of cooperating with them on some experiments. And one of the experiments here, I show you what it looks like. So typically, the flow cell is stainless steel. The single use is plastic. I'm not going to get into all the technical details about how they do the sterilization because I think that's still being worked out. But suffice it to say, this is very similar to a normal stainless steel flow cell, but it's made out of plastic. And it's going to come, according to what I've been told by Repligen engineers, shrink wrapped in a box that you can basically pull off the shelf, connect it to your VPX, and start your manufacturing process and get accurate data. And so we have been doing some comparison studies, and I cannot reveal the results of those studies, except we've had some positive results, and that these things do exist, these single-use flow cells, and they work. But they are not commercially available as of right now, and I think the development is still being completed. But you can see what we did in our lab. This is our pilot lab. To best assess it, we ran 2 VPXs simultaneously because we do have multiple ones in the lab. And with the 10 millimeter flow cell, which we typically use, that's the one we typically have connected in the pilot scale, and then tested at 22 millimeter, which is the size we use in the clinic, and compare the results. But like I said, we don't have the results to be able to show right now. Let me go to the next slide. So the known gaps and challenges to do the GMP tech transfer. So I think I mentioned before, the pilot scale lab we have is not GMP. It's like we make tox material, and it's the last step before GMP. So it is a really good place to test out these potential GMP technologies before you install it in GMP, because it's a 1/4 scale facility, and you can see what goes wrong. So the known gaps and challenges to getting this system from the pilot scale, from where we are now, into our GMP clinical facility. It's a little bit color coded. It's somewhat arbitrary, but I put the development of and the commercial availability of the single-use flow cell, probably the biggest gap in town, to it basically implementing this in the GMP space, because the other alternative would be to clean a stainless steel flow cell, and that would involve a cleaning validation, which anybody who's experienced with cleaning validation understands it's a very lengthy project to do, and so it's much easier if you can have the manufacture these single-use flow cells that you can just plug in. However, like I said, we know they exist, we've tested them, and so at some point, I assume that the kinks will be worked out, and they'll be able to certify these are fit for to be CFR compliant. The next one moving forward would be fitting the instrument into the UV-Vis skid. That's something that we've already mapped out in our facility. It's not actually too difficult in our particular case, but of course that's something individual to each manufacturing suite that will need to be done. System suitability testing, and that's something that I think that started to talk to Repligen about, and that's something that we need to come up with so that we're assured whenever a flow cell is installed, whether a single-use flow cell or a stainless steel flow cell that's been cleaned, how do you know that this is accurate? That's something that we'll need to provide an assurance that it is. The data infrastructure, that's something that I expect to be too difficult in the sense that in our pilot lab, we have already set up connections using an OPC UA data input output of the VPX, which is I didn't go into the features of the new software, which is Viper, but Viper is a software that was released to go with the release of the VPX hardware, and that is potentially a CFR compliant software that can have OPC UA, and that allows you to talk to digital. I'm trying to remember the exact term. The like a software like such as Delta V, a digital distribution system. There's another name for it. But in any case, that's how the data can get channeled in and streamed real time into this Delta V platform, which is basically the software that runs the skid and all the manufacturing operations. So that's obvious. Suffice it to say that you want to have your data stream with everything else that's coming in the dashboard, your pressures, the flow rates, and everything, and you can see it all simultaneously and potentially even control based on concentration. Electrical supply, that's nothing too serious, but it's something obviously consideration, because this is a sizable instrument that you're going to be putting on the floor. It takes up space, and part of that is GMP cabinet. So I won't go too much into that because that's something Repligen doesn't sell, but it's something that we have pursued because it's critical that an instrument in a GMP setting is going to be sealed to the elements because it can get splashed. Okay, so this is kind of the final slide I have here to sort of wrap up where we are and sort of where we're going in the future, without going into excessive amount of detail. But where we stand now is the FlowVPX is integrated into the skid unit operation so that the drug substance concentration can be measured throughout the UFDF in real time and potentially post-recovery in the pilot lab, as shown with real-time data monitoring. So I'll actually discuss this briefly. So you can measure with the FlowVPX using during UFDF, as you can see right here. That it sits there with the retentate. Then I didn't talk too much about it, but what happens then is once you're at a satisfactory UF2 set point, you do this thing called the recovery, which is literally basically gravity or pressure push everything, or with a buffer bolus, push everything out of the system into a recovery bag or vessel. Then what happens is there's steps where you basically take the material in the vessel, you pull a sample and you measure it, and you have to do all these things manually. And typically, the best way to do it now would be at line. And of course, like I said, unfortunately, in the clinical manufacturing, they have to do it offline. But ideally, you could do this also inline. So the issue with the recovery operation is that it's disconnected from the UFDF skid, because it's a separate sterile sort of operation in and of itself, and it's the pre-formulation. But what you can do, and this is something I don't even show the pictures, is set up a VPX system that would basically circulate the material that's been recovered in the loop and allow you to concentrate on using the inline readings to the proper formulated, or actually it's called purified drug substance, which is pre-formulated drug substance, and then even add the formulated materials, which, like I said, are the PS80 and the D and like something like EDTA, and then hit your final drug substance concentration. And as you can see, in this recovery thing, we had a loop which pumped through a separate VPX, and then after recovery, you're at a steady concentration. Then there's a dilution. They've hit their target concentration. Then they added their materials to formulate it, and that can all be done. Of course, this was done manually. The operations of dilution, but in the future, that could be done in an automated fashion. The key to it, of course, is that you need either a separate VPX instrument or something I thought of is that you could just basically take the VPX that's being used in the UFDF at the end of UFDF with a single-use flow cell, disconnect it, and then connect it to the loop that's on the recovery vessel with a separate flow cell. Because, like I said, unfortunately, these are separate operations, and you don't want actually cross-contamination. So what you would do is you would switch to a different flow cell. That would be probably a smaller flow cell because it would be in a loop with a pump. And like I said, we have done these experiments, and other companies have done these experiments. So basically, it's a way of measuring something in line that's in a bag or a vessel. And to get to where why that would be a big step forward in the UFDF process is on-the-fly adjustments during the UFDF process and product consistency. Actually, I'm moving backwards, sorry. What I'm saying is that these VPXs can be turned into real-time product monitoring and control devices, so that data feeding back to the Delta V can be used to control it in precise targeting of substance concentration. Because right now, that is all done manually by the operators. You're basically, using these things to hit a certain set point. And they have to figure that out based on mass. In the future, they could do it based on concentration. Well, actually, that's what they do now in the pilot scale lab. They watch based on concentration, but they still operate the valves and everything manually. The next step would be to basically set that up so that would be done automated in Delta V. And that's where we're going next. And then after recovery, you could be set up so that would be done all in automated fashion too by Delta V. So this has a potential significant increase in the process cadence, shortening the process by several hours by eliminating all those offline measurements and having a complete automation. But of course, like I said, this is a stepwise thing that will probably be the biotechnology will be moving through, as they move to increased automation of biologic manufacturing. Okay, so good timing. Yeah, so it took me about 45 minutes, it looks like. The FlowVPX development team, maybe just a few people here. So Lefa is my supervisor, and she's been very helpful. Danuka has contributed to the project. He's the PhD strategy lead at BMS. Julia is senior director. She's been involved with the project for quite several years now, doing some guiding. Melissa is downstream with Vivek, who's been helping me. Spencer is what they call the Manufacturing Sciences and Technology Group. Robert and Kevin are in the pilot lab, and they've been the ones who've been doing a lot of experiments along with Frederick, that were basically establishing the accuracy of those single-use flow cells and previously just the VPX in general. So Derek, Digvijay, and Danielle, they all left this year, unfortunately, but they were tremendously helpful in the previous 5 years that we worked on the VP and the VPX to advance this technology. Okay, so this is actually goes way back to 2017 when we started using this in our pilot lab and bench scale. And then I'll mention this, the last three is David, Salvera, Peter, and John, and they have been very helpful in getting this into the clinical space. So that's everything, and I guess hopefully everybody heard me, and that's everything I have. So go ahead and we'll ask questions now. Thank you.
Thank you, Jay. Great presentation, and we thank you for sharing that with our audience. Everybody, we're going to jump into our Q&A session. Just a quick reminder, if you have a question for Jay, please enter your question in the control panel in the questions tab. Okay, so let's see what we got. Okay, question 1. How has the FlowVPX improved your overall efficiency, and has it reduced redundancy / process time? How much is BMS tracking the improvements?
So we're not, so that's actually a little bit ahead of the curve now, because, well, I'll step back. In the pilot scale, it has helped actually quite a bit in that they're able to, so they've used the VPX or the VPE up until 2 years ago now, the VPX, to decide when to stop that for that UF2 concentration set point. When you're doing it by mass, so what happens is, like, say in the clinical manufacturer, they have to wait several hours to get this back. So in the pilot lab, they basically be able to shave like a whole almost a whole day off the operation, because they've got their inline VP VPX. They get the reading, they know when to stop the process right away because it's very accurate typically. Is what the VPX is. They take the sample, they walk it over to the solo VP just to confirm. Then once they know it, they do the recovery, and then they're good to go. And so another thing to emphasize too is that it's important to be it really makes the process more efficient if you can hit the UF2 set point very precisely, because then you don't have to do further dilution to get to your pre-formulation target zone, which is what's called the purified drug substance. You can literally basically stop your process, flush it, recover it, take one more sample. You'll find out it's basically in the correct concentration, because there is a bit of a buffer flush, so it does get diluted by having been recovered, and then you find out, right on target. You add your excipients and then go, and that's it. So actually, this is I do have a number here actually that I can cite, is that because they're using the VPE in the pilot lab, only I think a few times out of maybe out of about 30 of the last purification runs that they did for UFDF have they had to do another dilution to get to the targeted drug substance. Whereas in the clinical space, because they don't have inline monitoring, their UF2 is much more variable, so that their recovered material is much more variable, and they're having to do an additional dilution half the time. So what that means is that they have to add this step, which takes several hours and requires an offline 4 to 6 hour A280 measurement half the time. So basically, half the time they have to add half a day. So I hope that answers the question. But basically, it's saved about half a day on average for the pilot scale.
Wonderful.
Yeah.
Okay. Next question. What new insights has the FlowVPX provided into your process since your team has incorporated the technology?
One of them, I guess, is the mass is not a reliable way to measure concentration. I'm trying to think of the word, but basically it's a substitute, because, like I said, they do take a solo VPE measurement at the beginning of the process of the viral filtrated material, and from that, the change in mass of the system tells you what the approximate concentration should be. But however, because when you're going to you think about it, you're concentrating 20 times. So, and the holdup volume can be like up to several liters. And so they can calculate that, but it's hard to get a really exact calculation of holdup volume. So they have to factor that in along with the mass. So they get an approximate mass and an approximate concentration, and that actually is not that accurate. Usually it can be even up to like 10. When they've actually done a few comparison studies, it can be up to 10 to 15% off. So that's kind of, I think, what the one of the things that's been revealed, in, especially in the pilot scale, because we have this side by side data, is that it's you really at those highest concentrations, especially like I said, because there's a huge reduction in volume that you basically have the situation where errors can be magnified because smaller changes in mass will produce a large change in concentration at the when it's very concentrated versus when it's dilute.
Great. I I do like this one. Okay, next question. What would you recommend as a validation parameters when considering implementing the inline concentration methods such as the FlowPX during a UFDF process?
Yeah, so that's that one. Like, so validation is kind of a big deal. I wouldn't, and there's a lot to a validation, so I can't. I'll just say like one thing is that the one thing that you cannot deviate from, I think, is the 5% comparability. That it needs to be within 5% of the offline method, and that's a critical part of the validation. Otherwise, it could be validated. I think actually we did we've done a qualification inline and pseudo at line with the VPX. So when you get the VPX, there's this adapter that can be used that basically turns it into like a pseudo solo VPE. So it makes it much simpler to do a validation of a method. So that's something we actually did, and it passed the it's called a qualification, which is like similar to a validation, but not quite as rigorous. So we've done a qualification of this method with the VPX, both, like I said, using the adapter, which would mix it at line and inline, and it passed both of those things. So like I said, the most critical part that we were looking for, especially, was that 5% comparability across in a wide range of different concentrations, a very large dynamic range.
Okay. No. Next question. How did you align with other functions such as QC, AD, and MSAT to adopt inline VPT?
So that's something that's in progress, and I don't know if I could share too much because that's kind of a complex thing, and it's something we're going through right now. But I guess I'll say that we were kind of the genesis of the project, and I'm kind of, I guess, maybe proud that we were the beginnings of this PAT technology and process development analytics, because in the PAT there's the word analytical, and it's critical that people who actually, one of the problems in the early days of PAT, going back like when I was starting, I think, was that maybe a lack of analytical oversight and understanding when people the engineers, so-called engineers, or the chemical engineers who typically are the process guys or people, sorry, that were setting up the PAT instrumentation in the process without, like I said, a deep background in analytics. So I have, because like I said, I have a chemical engineering background. I've worked in Regeneron, I've worked in the process. I had that understanding of the process. I wasn't an expert, but I was enough that I understood, that these analytical instruments need to be incorporated into the process. They're not and be part of the process, and that it also has to have the same analytical rigor that an offline instrument would have. So that, so basically, I'm sort of taking a roundabout way to answer the question, but it really, the initial basically instrument development and basically like the trial, I guess, of the technology was done in process development and process development analytics specifically. And then what we successfully demonstrated this technology. Now it's actually that the other groups became interested because we established this as a viable technology. They first want some data, of course. So we got the data. We advanced it to this point where we well established that this is a viable tool, and now the other groups are coming in and basically connecting, and we're at the point, we've got several, many different stakeholders from many different groups that are sort of all kind of coming into the picture now, and kind of coalescing around our work. And we're sort of, distributing what we know to those partners, the stakeholders. But there's, like I said, it would be kind of complicated to describe all the groups, but the critical group that I'm working with now, especially, is the people in clinical manufacturing. I guess like the group, I don't know the exact name of the group because they change all the time, but they're responsible for new technology in the clinical space and the implementation of it.
Understood. Okay, next one. Do you see inline VPT ever being used for upstream applications?
Okay, so yeah, upstream. That's probably, yeah. This was something that I thought of before. So it's probably not in upstream, because you've got the so for biologics, especially monoclonal antibodies, you have the cellular material and you have the extracellular material, the media, and of course the monoclonal antibodies secreted in the media. So typically, if you're measuring, CQA, or which is like what the this instrument can do. Of course, it's like A280, or it's the UV-Vis, more correctly based. It can measure a wide range of different chemicals. However, you need to remove the cells first, and it's this because the matrix has so much other stuff in it. There'd be too much, I think, matrix interference to get, let's say, accurate concentration. However, once you get there is so this is not upstream. This is where it becomes downstream. But there are potential. I know the people at Repligen have been working on this, and others, that you can start using the VPX as soon as the protein, because some people have been using it for protein breakthrough. It's good enough for that. I think it's because that is where you at least have separated the protein from the matrix, and you can do a pretty accurate protein concentration measurement once it's been protein purified.
Okay, and this is good. Tell us how you got to where you are with inline VPT. What was your journey from inception to adoption?
Yeah, so that I think I mentioned it sort of during the talk. Really, the year I started getting into it was 2017, and it wasn't initially my doing. This was again, there was a person. I can't remember his name. Anyway, well, Derek Choi was one of them. I mentioned. I just want to give him some credit. He was the one who purchased the VPE instrument for his pilot space. And in terms, there was actually another guy involved, but I can't. It doesn't really matter. In any case, there was 2 instruments purchased for our process development group in 2017, and that was by like I said, someone in process development and someone in the pilot lab. So I started working at the time because they wanted to know the accuracy of these instruments. So we started doing tests for them, to basically comparability testing, and basically coming up with ways to do system stability, accuracy testing, and sort of just generally taking ownership of the instrument. And then I actually started getting involved in the solo VPE, because that was actually right around that time that we started working on these higher concentration processes. So as you can imagine, like to measure something 150 to 200 grams per liter using a cuvette is a real pain. So we were at that point, we really wanted to start testing more and more with the solo VPE. It's so much easier at those high concentrations. So that, I guess, is like the journey we started. Like I said, 2017, 2018, when I was sort of put in charge of the flow and the solo VPE projects, and basically helping, and especially because they were a little bit unusual because they were PAT projects, and we didn't really have any PAT at the time. And so the PAT initially was just to accelerate process development, but now it's, of course, it's not just accelerating process development, but potentially accelerating manufacturing.
Wonderful.
Sure.
It looks like all the questions have come in. This concludes our Go with the Flow 2 session with Jay West. Thank you all for attending. If you have any additional questions, you can reach us after the presentation. We'll be happy to answer any questions that you have. Thank you all for your time. Look forward to the next round.